Susumu Tonegawa to start off our MENDEL lecture series for 2023. Susumu was born in Japan and graduated from Kyoto University in 1963, and then went to the US for a Ph.D. at the University of California, San Diego. And then he went to the Salk Institute and did a postdoc with Renato Del Becco of the media fame, and began learning immunology. And then he went to the Basel Institute of Immunology, which was being set up by Hoffmann-La Roche in 1971. And he began to work on antibody rearrangement, antibody gene rearrangements. And he continued doing that in Basel until 1981, when he moved to MIT as a professor. And the antibody work came to a conclusion around 1983 or so, and he won the Nobel Prize in 1987 for that, and the Lasker Award in the same year. And then he was also given a cultural award by the Emperor of Japan. I think this is also what they call the National Treasure Award in Japan. So after the Nobel Prize, he switched fields. I don't know if that's correct. He switched fields and began to work on memory in the brain, real memory, not immune memory, and developed techniques for marking the cells that are activated when a memory is formed and that get reactivated afterwards. And the thing is called an engram. It's the trace of activity in the brain that you reactivate for memory. So in this capacity, he switched to being a professor of learning and memory at MIT, and around him they developed then this Picower Institute for Learning and Memory, which is still at MIT, and he's still a professor there, though no longer director, and he is also a Howard Hughes investigator. I think that is most of it. So it's my pleasure, there are other things, but it's my pleasure to present you with the MENDEL Medal for this talk. Please. Yes.
Oh, thank you. Thank you very much. Oh.
I'm in trouble. There
Wow, wow.
Yes.
That's great. Yeah. Thank you very much.
Very good.
I can, yeah.
Back there.
All right. So I have to put it somewhere. Okay. Okay, thank you for the nice introduction. And I have to start with a little bit of apology. You see this title, it's slightly different from the one I announced 2 months ago. When I was assembling all these slides, I realized to try to cover both immunology and neuroscience is too ambitious. So because this is a MENDEL lecture, who is the father of genetics, I thought it's better if I focus on topic that is more closely related to what Mendel discovered. Okay, so let me. I will mention at the end, just using a few slides about the neuroscience, learning and memory we are studying these days. Okay, so that said, when early 1960, 62 actually, I was, as I was introduced, I was a student, the senior student of the Department of Chemistry of Kyoto University. You know Kyoto. Kyoto is the old capital of Japan. And I was trying to decide what I'm going to do after I finish, after I graduate from Kyoto University. And one of my friends told me that there's some new biology is erupting in Europe and the United States. And that's called molecular biology. Now, I had not been interested in this classical biology, but since this sounds very interesting, I started reading some papers, and I decided that this is the field I'm going to be trained as a graduate student and stay in research in this field. But there was a problem. Problem is there was a molecular biology is so new. There was no place in Japan the lab carry out this type of research. So I have to go abroad, Europe or US, and I was very fortunate. One of my professors knew someone in San Diego that he is creating a new department of molecular biology at UC San Diego, UCSD. So he wrote a letter for me, and I was amazed that I was accepted. So in '63, the summer, I left Japan and joined in the graduate school in UCSD. You know, the campus is in the beautiful town, or northern town of San Diego called La Jolla. Probably some of you have been there. And at that time, it was a rather quiet place, but the institute, that campus was the university, newly created university, was borrowing the laboratory of the Scripps oceanography. And that research institution was quite famous. And this is the peer of the Scripps Institute, where the this pier sticks out in the Pacific Ocean on the beautiful California sky. Now, their department was very small. There are only 5 professors, and then there's only one more layer of graduate students above us. Okay, so but they were all excited about this new research direction. So they were very active, and I very much enjoyed having the thesis work there, taking about 5 years. Now, at that time, so it is generally agreed that so-called molecular biology started with this Watson-Crick's, the elucidation of double-stranded DNA. Okay, this was 1952, actually, quite almost 10 years earlier than that period I'm talking about. And this work was succeeded by a series of major discoveries in what is called molecular biology. And this one shows this is a Messelson-Starr's experiment demonstrating the gene, DNA, is transmitted to the next generation of bacteria, E. coli, by what is called semi-conservative replication. And that was a very important finding, and the experiment was very neat. I don't want to explain in detail, but it was considered to be a major finding, which was 1958. And then came another big discovery, which was carried out in Paris, Pasteur Institute, by this, do I have any pointer here? Okay, I forgot about the pointer. The one on the left is the Jacob, Francois Jacob. Middle is the Monod, Jacques Monod, okay? And Dwarf is their sort of mentor. And these people, oh, thank you. Oh, this I can use, okay. Okay, so now, Jacob and Monod, for the first time, proposed this credible mechanism of how gene is expressed to RNA first, and that is translated into proteins, as shown here. So this RNA was not found yet, but they postulated there will be special kind of RNA, messenger RNA, mRNA. And the expression of the gene, which is clustered, which they call it operon, is regulated by another hypothetical protein called repressor, okay, which will bind to the upstream of this gene cluster and regulate level of expression of DNA, okay, into mRNA, but providing same genetic sequence from DNA to RNA, and that is translated into a peptide amino acid sequence. So that is called the operon hypothesis. And I was really intrigued by this finding. So this was quite instrumental for my decision to go into molecular biology. And then a few years later, it was still in the 60s, Marshall Nirenberg and Goblin Corona identified the genetic code for all 20 amino acids. You need a code to convert this information encoded in a nucleotide sequence into a specific amino acid sequence. So they, using another clever experiment of cell-free protein synthesis system, they identified all the code for the all 20 amino acids. And finally, putting all these together, what is called the central dogma of molecular biology was laid out. Francis Crick and some others together, and the DNA will replicate, and then information in DNA is transcribed into messenger RNA, and that information is translated. They call it translated into polypeptide chain. That is the central dogma of molecular biology. So, as I mentioned, I just go through very quickly again, the same thing. Start with Watson-Crick, Methylson-Stahl, and Jacob Monod, and Brenner, Jacob, Watson identified the messenger RNA as a real thing. And Nirenberg and Corona identified the genetic codes. And finally, late 50s and early 60s, Crick and several other people together came up with this central dogma of molecular biology. Now, as you can see here, these were all done in the last 2 years of 50s, and then first year of 60s. Okay? And in fact, around the time that I finished Ph.D., which was 1968, and in fact, many of these people, except Methelson and Starr, had already received the Nobel Prize, Nobel Prizes, okay? So, although I enjoyed very much learning about the early stage of, early phase of molecular biology, the question is, what are we going to do when we have our own lab? I mean, all these major things is already discovered. And only thing we could think of, I could think of, is to go to more complex system. This work is all done by prokaryotic, simple prokaryotic system, E. coli and viruses which infect them, which is called a phage. Okay? But, you know, it was not technology, the principle would also apply to eukaryotic system, at least that's what everybody believed. But when it comes to the detail of eukaryotic system phenomena, such as ontogeny, developmental of the single organism, maybe cancerous transformation of eukaryotic cell, replication, regulation, and all of those things is still unknown in eukaryotic system. However, the technology which will allow you to do this very precise experiment with animal cells was very scant, okay, not around. So there was a problem. The question is there, but there's not enough technology. So the only thing I and my friends, when we were trying to graduate, was that we go to some intermediate system where, for instance, one can use the animal cells, but you try to grow them outside of animal, tissue culture, okay? Tissue culture of, for instance, embryo cells. And then you infect them with some virus, which will change the phenotype of those cells. And then, you know, it's a cancerous transformation. And maybe this is a system where it is sort of good for transition from prokaryotic to eukaryotic system, particularly if you use small DNA viruses like polyoma and SB40. Then you can isolate the DNA, fractionate them, and all those things. So I was trying to find a lab like that who are trying to do this type of transition study from prokaryote to eukaryotes. And fortunately, I found out just right across the street from UCSD campus, there was a new institute built that called Salk Institute. And that Salk Institute, which is showing this beautiful campus of a courtyard of Salk Institute, overlooking, again, Pacific Ocean. And in this institute, there was a professor whose name is Renato Dalbecco, who was exactly working on this kind of a hybrid system. Okay? So I applied, and I was accepted. Okay? So I was further trained in the early stage of eukaryotic molecular biology. However, there was a problem. This time, the problem is I went to USA as a graduate student using a visa called J-1 visa. This is a notorious US government visa which require you to leave United States after 1.5 years of a postdoc. Okay, you have to go out. And this was very strict at that time. I think it's quite strict now too, but there may be some exceptions these days. But at that time it was very strict. So I had to find a place to go. I didn't want to give up what I have learned, and I was quite still inspired to make some significant discovery in this field, in eukaryotic molecular biology. So I tried a few places to have a shelter of 2 years. If you stay out 2 years from United States, you can come back to US. So I looked at Canada, I looked at the Japan, but none of them was good enough for me. And it was about two weeks before I was going to be deported from the US. I received an airmail letter. Airmail, there was no internet then. So people have to communicate either by international phone call or airmail letters. I don't know, you guys, older people would know, but the younger people may never seen it. But it seemed stationary because airmail is charged by weight. So they use as thin paper as possible, and you write with a pen what you want to say. And I received a letter totally unexpectedly from my professor, Renato Dalbecco. And the letter said, "Dear Susumu, I don't know where you decided to go when you have to leave my lab. But if you have not entirely decided yet," it said, "I have an additional suggestion." He doesn't say you have to do what I suggest, just I offer you additional suggestion. And in Basel, Switzerland, there is a new Immunology Institute is being formed. And they seem to have already assembled very good immunologists, but they don't have any molecular biologists. And then he says, I believe, that him, Renato believes, if someone like me who is trained well in molecular biology is willing to go into immunology, the person may have a good time. Okay, that's all it says. And I thought this is a crazy idea, and I asked around my colleague postdocs, and everybody said, "Oh, that's crazy. Renato doesn't know anything about immunology." And so I put a letter in my drawer, and I never did anything. In fact, the letter also said if I'm interested in, I could write a letter to the chairman, to the director of this institute, Niels Janne. But I didn't write it. And then he came back 2 or 3 weeks later, and he came to my desk and said, Susumu, did you write a letter to Niels? So I said, no, I don't know anything about the immunology. I asked around the people, they didn't think that's a good idea. So I decided not to take that advice. And he said, that's too bad. I thought that's a good idea for you. And then he left. So I thought about it again. And then the thing is, I was quite impressed before things like this happened, how insightful he is in looking into the future of biology. As I told you, he was already working sort of interface of eukaryote and prokaryote biology. And so I believed, I decided to gamble to listen to his advice and decided to go. So in the winter, the problem is that I didn't even know where the city Basel. I never heard of such a city in Europe. Never heard of Niels Jane, immunologist. So it was not very encouraging to make this decision, but I did. And here the front of a small institute, Basel Institute of Immunology. Okay, and this institute was established not by Basel City or a company, but it was built by owner of a drug company called Hoffmann La Roche. Okay, I mean she was a billionaire, and she just used her money, her private money, to build this institute. Because so that she can enjoy interacting with the scientist once in a while. And I went there, and it was very good institute in terms of, for instance, funding. There was not really budget. I mean, you can just use whatever you want to. And also, Jane's policy was anybody who have a PhD, whether you have a postdoc experience or not, if they want, they can be independent. They can have a lab, maybe half of a lab, but one technician. Okay, so you can start doing what you want to do, independent from your boss, with your technician. Okay, it was like that. And the first 2 years, I thought I did think I made a mistake by coming there. Because I go to the seminars, and I don't know what they are talking about at all. I could not understand anything what they are talking about. But I think I remember sort of beginning of a third year, I learned there has been century-old major mystery in immunology, okay? And that is there unresolved. And so in order to explain that to you, let me just give you a couple of slides to remind you the major player of the immune system, that is the antibody, okay? So here is a structure, schematic diagram of antibody molecule made up with two light chain, two light chain, identical light chain, and two heavy chains. Okay, they are disulfide bonded. And then what is striking is actually the amino acid. If you look at the amino acid sequence, some of that was available because there were tumors of antibody producing cell called myeloma was available in a different clones of myeloma. And if you look at you can sequence the antibody, which is generated by each of these myeloma clones. And every time they sequence amino terminal area of light chain and heavy chain, the red area, sequence is different from one to another. Okay. On the other hand, if you look at the rest of the sequence here, shown in blue, sequence is variability is extremely limited. For light chain, there are only actually 3 types of sequence. Heavy chain, there are only 6 or 7 types. The tremendous variability difference between the amino acid side and the carboxyl terminal side. So, on the other hand, people started finding how these antibody molecule bind to antigen molecule. And it turned out it is a variable region which determines the affinity of antibody, which is shown here. This is the light chain variable region here, heavy chain variable region. And here is the lysosome protein antigen. Okay? So there, it is a variable region which recognize and bind to antigen. And so antigen antibody relationship structurally is like a key and lock hole. So they have to fit very well in order to for antibody molecule to initiate the process of getting rid of antigen. Okay, so since variable region sequence is so different, there was a big debate about how the total set of antibodies are in fact encoded in the genome. Okay. And so that's the problem with this God, they call it God mystery, G-O-D mystery. So question is, how does the immune system handle the enormous diversity of antigen if there has to be an antibody gene for each antigen? Okay? Because antigen variability that immune system can deal with is really amazing. If the chemist prepare one new compound, never existed on the planet, and then put that together with a big protein and inject to rabbit or a mouse or whatever, they make antibody. Okay, so this antigen has never existed, and yet antibody that have sufficient affinity to synthetic antigen can be formed. Okay. So there are people, however, 2 school of people who try to explain this this mystery, and one group is called germ liners. Okay, actually, this is not a great theory. It's just stating what molecular biology had already told the people. That is, all antibody gene present in the germline genome, whatever variable it is, the gene product is, and transmitted as such to the progeny. Okay, there's nothing special about these genes compared to all other genes we have. Now, but there are some other people, particularly those people who know this problem of God, as well as have some understanding of genetics. They want to think there must be a limited number of germline genes, not for every antigens, but a limited number of germline genes that are diversified during the ontogeny of each organism. In other words, your antibody repertoire, you create yourself during develop your immune system. Okay? So that they are called somaticians versus germline. And they argued when I went to some immunology conference, they were always arguing which one is correct. So this article, I found it when I was preparing these slides. This is a short review written by this issue of antibody diversity. And the title is it all coded for the germline? Okay, that's the title. Written by Hans Wichtel. Hans Wichtel was a well-respected immunologist. And he was, in fact, that turned out to be, he's also the chairman of the committee, the Karolinska Institute committee, who gave physiology, medicine, Nobel Prize for several years. Okay? And you read this red part, probably you have done that already. In the prejudiced mind of the author, Mr. Hans Wichtel, the new data strongly support germline theory, leaving little, if any, room of somatic mutation to play any role during ontogeny for immunoglobulin genes, antibody genes. So that was 1973. That was around the time I was starting work on this problem. Okay, so there was tremendous wind against the idea of there is a somatic diversification of antibody gene. Around that time, a couple of people at Caltech, Dreyer and Bennett, in order to explain this enormous difference in amino acid sequence diversity in the variable region versus the constant region, they came up with this, what I thought, or many people thought, a very ad hoc hypothesis. That is, in fact, in the genome, there are a number of V-region coding genes on their own. And there is a single C gene for one particular type of light chain or heavy chain. And there is some kind of linking between those V gene, one of the V genes, thousands of, maybe tens of thousands of V genes are picked up by single C gene, and they get joined. Okay, and so this was not very seriously taken by molecular biologists because this is against the one gene, one polypeptide chain hypothesis, which is one of the fundamental principles of the dogma of molecular biology. But it was there, and the idea was there when I heard first time the Hamilton Smith and Daniel Nathan's discovery of the first restriction enzyme. It immediately hit me that one can test at least where Dreyer-Bennett hypothesis is correct or not, if there is any kind of somatic changes, the combination going on between V and C genes. So I don't want to give you too much detail on this, but I just explain to you what was that idea, my idea. So you use myeloma, single myeloma clone. They produce, as I said, single immunoglobulin, okay? One heavy chain, one kind of heavy chain, one kind of light chain, okay? So I use that as the source of genomic DNA. You extract DNA, and then you digest them with a restriction enzyme, the one which would cut relatively infrequently 6 nucleotide recognition type, like ECHO R1 or BAM-H1 or things like that, okay? And these restriction site is scattered in entire genome randomly, okay? Now, we can also isolate genomic DNA from non-immunoglobulin producing cells, such as a very early embryo cell. I mean, I'm talking about a mouse here, not a human, mouse, okay? And you just in parallel digest with the same enzyme, and you fractionate this in some electrophoretic method, okay? And then if we have a way to purify immunoglobulin messenger RNA probe, particularly if we can produce not only hypothetical V plus C messenger RNA, but also C messenger RNA separately, okay? And then you can hybridize all these DNA from each fraction of the fraction of electrophoresis. And my logic was there must be a difference in the hybridization pattern. If you look at along the sides of DNA that are fractionated by electrophoresis. Do you get the idea? Okay. This is important, otherwise what I show you mean nothing. Okay, anyway, so that was the idea. So what I had to do was to find a way to make this hybridized RNA probes to hybridize. There is the restriction enzyme digested the DNA population. And here the way to do messenger RNA purification, and we could design this preparation so that you purify V plus C gene probe as well as only C gene probe. Okay, and then we carried out experiment. And this is what we got. And this is a little complicated slide. There are four curves here. X-axis is a migration of DNA, so it's fractionated by size. The one on the left is a bigger fragment. The one on the right is the smaller fragments. And then each fraction was isolated and hybridized to two different kinds of probes, whole messenger RNA probe and 3' messenger RNA probe. And this was carried out for myeloma DNA and embryo DNA in parallel. So there are 4 different curves here. So to make it easy to read this, I asked my postdoc, Michele, to fractionate, to separate the curves obtained by embryo DNA on top versus myeloma DNA at the bottom. But each one fraction is still hybridized to C gene probe or V plus C gene probe. And then the most important thing, message from this, is that the hybrid, whether you use the V plus C probe or C only probe, hybridization pattern of embryo DNA and the myeloma DNA is entirely different. You understand what I mean, right? Okay, so this is, yeah, so here the interpretation of this, okay, of this finding. And interpretation is there is a DNA fragment in the genome, which is flanked by this BAM-H1, nearest BAM-H1 restriction site, and it's a certain site here, okay? And there's a C gene coding sequence somewhere in the genome, and that is also flanked by BAM-H1. And this side of V fragment, C fragment are different because there is no reason to be the same in terms of distribution of BAM-H1 site. And if these V to C gene joins by recombination, you get a fragment like this. And that size will be also different from this or different from that. And the sequence in between them in the germline genome, God knows how long DNA fragment that is, will be squeezed out by recombination event. Okay? And it gets lost from the cell lineage. So that's why you get a very different hybridization pattern. So this is the first time that somatic generation of antibody diversity by recombination, somatic recombination, was demonstrated. Okay. Now, this work was carried out in 1976, and it was enormously tedious work because at that time we didn't still have what is called the southern blot technique. You know, some of you probably know the southern blot technique, and if we use that technology to fractionate the DNA, then let nitrous cells filter bind to the fractionated DNA. Then I got 10 microgram of DNA is sufficient to carry out that experiment. But if you want to do manually the whole thing like we did, you need more than 5 milligrams of DNA. Okay? So it's a very laborious experiment. In any case, this was demonstrated first time when we used the kappa, looked at the kappa locus. But at that time, there was a second wave of molecular biology appropriate for study of eukaryotic system was coming out. And one I already mentioned, Smith and Nathan had a restriction enzyme discovered 1971. And then southern blot came 2 years later. And then the California people, Paul Berg, Boyer, and Cohen, invented this recombinant DNA technology and gene cloning, okay, from a eukaryotic source. And then Ron Davis also invented a method called R-loop mapping for cloned DNA. I'll mention that too in a minute. And then Maxim Gilbert and Sanger around the same time, maybe a year apart, had their own method for DNA sequencing. So this is exactly the time when I was working on our project. So as soon as that we got to know this new technology, we could adopt that and to do the analysis at the final level. Okay? That's exactly what we did. And the first thing we did is we confirmed there is somatic VC joining in a lambda chain right system. And what is shown here on the left is the lambda chain producing myeloma DNA, and then next is the embryo DNA. You see the one band is missing in the embryo DNA, and that's this one, no, this one here, and that is hybridized both V plus V region probe and the C region probe, okay? And that is the C lambda, there is a diploid. So rearrangement occurred only one of the two chromosomes. So the other chromosome stayed in the germline state. That's why you get the germline C and germline V2 also, okay? Now, interesting thing is the embryo doesn't have any V plus C fragment. And if you look at kappa-producing myeloma, also you don't find the V plus C DNA fragment, which is not surprising. It turned out that this VC joining apparently is so specific to particular gene locus type. So kappa myeloma produces kappa chain, but not a lambda chain. And therefore, there is no V plus C lambda DNA fragment detected by southern blot. I hope you understood it. Now, we went on to clone all these fragment DNA, genomic DNA fragment, V plus C lambda, and V1 lambda, and C lambda, 3 clones, unrearranged and rearranged, okay? And then we try to map the location of V plus lambda, the transcript. And for this purpose, there is a very nice procedure which Ron Davis invented. That is, you take a double-stranded cloned DNA, and then you mix that with the messenger RNA, whose gene are supposed to be on that DNA fragment. You mix them, and you heat this up only to 70 degrees with a high salt. And then what happened is the DNA duplex will come apart, and then if there is a complementary RNA in the mixture, messenger RNA will replace one of the DNA strands. So you get a DNA strand messenger RNA hybrid and a single-stranded DNA in that part of the DNA fragment. And that is called R loop. But what we found is this, which shows that the triple R loop. This was not expected from the idea that the gene is contiguous all the way through. Okay, so the interpretation of this is in here, and here is the V plus V gene. Here the C gene. This is the rearranged gene, but it turned out they don't get rearranged, but they are not contiguous. There are some sequence in between that does not end up in the messenger RNA. So this is the first demonstration of the presence of intron, what we now call intron, in a eukaryotic system. Only before that, a few months before, with the adenovirus, Phil Sharp and Rich Roberts discovered what is called split genes. Okay. But as far as the eukaryotic genome is concerned, this was the first one. And here, C gene mRNA hybridized here, V gene mRNA hybridized here, and the DNA in between, a double-stranded, is squeezed out. Therefore, you get this triple RNA loop. Okay? Now, then we went on. It's a little bit more complex. We went on to sequence all these relevant regions of these 3 different DNA fragments. Okay? One is the germline V gene, and then second is myeloma-derived rearranged V plus C gene fragment. And actually, third is a germline-derived C gene carrying fragment. Okay, and I didn't show the sequence itself, but for schematically, the homology of the sequence between these 3 cloned DNA fragments is shown here. Okay, so you start from the upstream side, and 2 hours comparing germline V and myeloma V plus C, and to our surprise, even before V gene mature V peptide coding sequence, there was a short intron. Okay, and there is a small DNA sequence that encode what is called a signal peptide. The signal peptide, probably some of you know, that is one which is required when a protein is made into plasmic reticulum and has to pass through the membrane, and it is a signal peptide which allow them to pass through it. Okay, and it turned out signal peptide is between signal peptide and the mature variable region coding sequence, there is another second intron, short intron. And then if you compare the homology between V gene and the V plus C gene coming from myeloma, sequence is very homologous, okay, until it hit their triplet. That is prematurely, where homology is prematurely disappeared between germline V plus rearranged myeloma V plus C, V plus C gene. And it turned out there is a short peptide in V and C joint region, but within the V region, it encoded yet another segment, DNA segment, which is located not close to the variable region coding sequence, but the one which is linked to C gene coding sequence by this non-large intron. Okay. And recombination occurred here between V and J. We call that short peptide, which is supposed to belong to V region, J peptide. The recombination occurred in here. Okay. And there was this was found only by determining a nucleotide sequence. You could not find that by the R-loop study. Now, using this information, you can schematically draw what is going on with lambda immunoglobulin genes. You start with a signal peptide, exon, short intron, V region coding exons, and then somewhere else in the same chromosome, there is a short J coding exon and the big intron and the C coding exon. And then in the myeloma cells, there is a recombination, precise recombination occurred of V and J. Okay, not V and C, but V and J. And this is joined, and now this is transcribed to the pre-messenger RNA, as indicated in here, and it goes out of the nucleus, and it goes to RNA splicing mechanism get turned on, and then the intron is removed, both of them, and then that that is a peptide that from which signal peptide is also removed once it goes through the membrane of endoplasmic reticulum. So this is the whole process that we worked out with a simple lambda chain gene locus. Now, when we did a similar analysis with a more complex variable, the kappa gene sequence locus, mouse, this is just fortuitous, but in case of a mouse, light chain is a kappa type or lambda type, which is defined by C region sequence, but more than 95% of mouse lambda chain are the kappa type. Only less than 3% is the lambda type. So so corresponding that, then the arrangement of gene segment in the genome is much simpler in the lambda compared to kappa. If you go to kappa, then you find something like this. Here the signal, there are about 200 to 300 V gene coding sequence clustered in the genome, okay? And that is followed by that is followed by 5 different J segments, not a single one, like a lambda locus. And that is followed the C gene sequence. And each myeloma, each B lymphocyte clone, there is a random combination of V gene, one of these V genes, and one of those 5 J segments. So this combination already generates some additional variability compared to the variability already existing in the germline by the somatic process. Okay, because in some clone, as indicated, V3 can join with the J3, but in another clone, V3 can join with the J1. And this combination will generate additional diversity. Now, if you go to heavy chain gene, there was even another surprise to increase the diversity by somatic recombination. That is the variable region gene, variable region peptide was encoded not to 2 different segments, VJ, but another VDJ. Okay? And there are something like 25 D segments, which are different in sequence. And then for the for the kappa chain gene, this is the, did I go to heavy chain? Did I skip it? Yeah, it looks like I think, yeah. So this is the, this is the kappa that I described. Now I'm talking about the heavy chain. Oh, no, in case of a heavy chain, there's even more 3rd DNA segment, which also contribute to variability in the variable region, VDJ. Okay. And again, there is a random recombination between V and D and the D and J, and therefore to further amplify the somatic method of diversifying the final product. And on top of it, I don't know, this may be too detailed, but it turned out when J gene segment joins the segment at the joint at the JH segment, in each event of recombination is not exactly the same place. It shifts upstream, downstream by up to like 3 amino acids. Okay. So that's additional source of diversification. And all together, here is one can calculate how much diversity one can generate by somatic recombination. And as you can see here, heavy chain, 300 germline B, about 20 D, and 4J. You multiply this, then you get 24,000 variability. And then light chain, 300 and 4, 1,200. But then heavy chain and the light chain joins to some extent randomly, okay, to make one antibody. So now you can multiply this, and you get 30 million different, possibly 30 million different variable region sequence. And starting with less than 1,000 gene segment that is inherited. So this made it clear that the somatic recombination is a crucial mechanism for diversifying germline inherited immunoglobulin gene sequence. But so far, I have talked about only the diversification based on the recombination, DNA recombination. But it turned out, as the study advanced, more and more nucleotide sequence of germline, the V region, and rearranged embryonic V region sequence was determined, nucleotide sequence. And you look at the compare, you notice there are single base changes, as you can see here, for the right chain, and here's the heavy chain, V region, okay? And you can see they are clustered, more or less, in the area of the variable region, which is involved in the direct interaction with the antigen. It's complementary determining residue areas, okay? So it looks like there is a somatic hypermutation is also going on during the development of B lymphocytes. This you cannot explain by the recombination, because it's a change, it's a very, most of the cases it's a single base change, okay? So now, what's the purpose of having these two major mechanisms to diversify germline inherited immunoglobulin gene? And that became very clear when the molecular biological study was combined with McFarlane Barnett, all, oh, sorry, I don't like this. All the clonal selection theory, okay? So lymphocyte, in this case B lymphocyte, bone marrow-derived lymphocyte, start with stem cells. Okay, it doesn't express any immunoglobulin yet. But as it differentiate and generate B lymphocyte, bone marrow-derived lymphocyte, there is each clone express one immunoglobulin polypeptide chain as a cell surface receptor, okay? As shown, yeah, as meant to be indicated by this immunoglobulin of different color or different shape, okay? Now, that's where your immune system waits for the invasion of a microorganism, antigen. So antigen comes in, and what happened is one of those lymphocyte clone that happened to have immunoglobulin receptor on the surface who fit well with that particular antigen will be selectively stimulated, because this antigen-antibody interaction, antigen receptor interaction triggers the way to proliferate, okay? And therefore, they divide and also differentiate into what is called a plasma cell. That is a factory for making that particular selected immunoglobulin as an antibody. So that is what is called a primary immune response. So you have a COVID vaccine, and in the first shot, what you get is this. It's not very, you have some immunity, okay, by primary immune response. However, it is usually not strong enough. Therefore, you have to get a boosting, okay? You have to get vaccinated again. And so here's the explanation. So this part, this part is the same as the previous slide, okay? This is to generate a primary response. But when these cells differentiate, these B cells differentiate, there is another pathway reserved for different kind of differentiation. That is to generate what is called the memory B cell, okay? And in there, during this pathway, somatic hypermutation machinery is activated. It is designed that way, okay? Therefore, many of the immunoglobulin clones that was initially selected by this will mutate, receiving single base change here and there in the variable region. And most of them will be lost because they usually have, most of them have a lower affinity than original antibody because the mutation is random, okay? But some of them had produced antibody that is even higher affinity than original antibody here. So this is what I meant by having a little bit of a different shape of immunoglobulin receptor, which are generated from this pathway, memory cell pathway. And when the secondary response takes place, these are the immunoglobulin clone, B cell clone, that has even higher affinity than primary immune response. It selectively proliferate, will selectively proliferate and differentiate into plasma cell. So that is the process of secondary or tertiary immune response. And they have a higher affinity. And then those which did not, which mutated in this process improperly, not for higher affinity, get lost because they don't get stimulated by low concentration of antigens still around in the system. Did you get it? Okay. So this is amazing. I mean, this is almost God-made scheme to me. But this is what is happening, actually. You can tell by doing various experiments. And now I go back to this how germline sequence is diversified calculation again. Now I include not only recombination, but also quasi-random pairing, somatic hyper mutation, the item number B, okay? And you put them all together. Remember, without any somatic mutation, we got like a maximum of 10 million diversification. But now we can go even 1,000 more, fold more, 1 billion, more than 1 billion diversification. Basically, every lymphocyte you have had in a secondary response have the different antibody in a single cell level, 10 to 9. So that's the way it is. So I'm finishing this, or is it already, I passed 10 minutes. But let me just tell you very quickly 2 other things that I'm not going to explain in detail, but this study also generated 2 other important things about expression control of immunoglobulin gene. That is, number 1 is that here the, wait a minute. Okay, this slide is a little bit screwed up. So forget about this colored part. This one here, just say E. E stands for enhancer. There is something called enhancer. Probably some of you know. That is the additional mechanism with which that you can regulate expression level of genes into mRNA. Okay, and remember here the V gene here, and then D. This is the heavy chain case, J segment. So entire heavy chain locus in the chromosome, there is only single enhancer element. Okay. There is hundreds of V genes and so on, and also downstream there is a further recombination takes place within the constant region from IgM type, IgG type. Secondary immune response is mostly IgG type, which is defined by the constant region heavy chain, as opposed to IgM type, which is the antibody immunoglobulin you find in a primary response. Okay, so there are two kinds of recombination is going on. One is the VDJ recombination, and second is from primary to secondary response, there is a mu gamma recombination with a different type of recombination. But there is only in the entire locus, there is a single enhancer element. So that allows the V genes which happen to be selected for VDJ joining in case of a heavy chain and come very close to the enhancer element, their expression will be boosted up. Okay, and that's what we need in order to make very high level of immunoglobulin antibody. And then we found that enhancer element, I think 1983, and there were also people in Zurich, Walter Schaffner's group at the same time. The two papers are published in the same Cell journal because they happen to be really same time. And this is the first enhancer element ever identified among all other genes for eukaryotic genome. Okay. So immunoglobulin studies, my primary purpose was to cope with antigen diversity, but actually it also contributed more general manner for eukaryotic gene expression control by having this things like enhancer element in the genome, not just a virus, but a eukaryotic gene in general. Okay, and finally, maybe I'm taking too long. That's what I was afraid. Finally, immune system. I only talked about half of the immune system. The other half is carried out by T lymphocyte instead of B lymphocyte. Okay, T lymphocytes do their own thing, such as immune response, cellular immune response, cell-cell interaction immune response, or even regulation of a B-cell response. That was known. But the thing that is equivalent to immunoglobulin cell surface receptor was not known for T cell for a long time. And we could not find those receptors as a protein. But you know how it was found? It was found by finding their DNA first, instead of finding the protein and then find the DNA genes. And this was done by something called differential hybridization, subtraction hybridization technique, and I will not go into the detail. So this was also carried out around that time, 1984, by two groups, our group and three groups, and Ron Davis and another group. So I stop here. Okay, that's all. But I just summarize by here. So remember, we were looking for solution for God. Okay, so amazing solution for God. That is, the immune system is microcosmos. Your immune system is your microcosmos as opposed to real cosmos, small, very tiny cosmos. That for which Darwinian evolution, which takes place within a single animal, yourself, or mouse, single mouse, this Darwinian evolution takes place during ontogeny, not millions of years of real evolution of organism. Okay? It is happening in your body, in your immune system. So antibody and the T-cell receptor genes undergo random recombination and mutations in your body while you are your body is developing, and those that fit better to the environment, in this case antigen is environment, it comes from environment, are selected by them. So our system uses our enemy as the selector of the better antibody. Do you understand? I mean, antigen is more or less like for us, microorganism antigen, COVID antigen is our enemy for survival. But we are using them to let them select a better fitting antibody and T-cell receptors by letting the antigen select for preferential proliferation of B cell and the T cell. Did you get the idea? So that's all I want to say. And I think it was worthwhile. We had a great time during these 10 years of research. And I want to, although it's getting too long, but I can't acknowledge all people who contributed to this work, because I counted when I was making slides, there are at least 60 people, mostly Basel time, but somewhere after I moved to MIT. And if I do that, I'm sure I will misspell some names. So I decided not to type it. But I also want to thank especially to Renato Dalbecco for the suggestion to plunge into immunology. Without it, I could not have had this amazing adventure. So I don't have time, but so I would not talk about our current work on the neuroscience system. If you want to ask about it after my talk, you can come to me. But that's it. So before you say, I have one little thing. I have to confess, I have a hearing problem. I have a hearing aids, but sometimes if you talk from the back, ask questions, I may miss some words and I won't be able to understand the question. So if you don't mind, if you are sitting in the back, please come closer and ask me in front of me. Otherwise, hopefully some people will repeat your questions. So I'm sorry about this inconvenience.
I can try repeat questions if that helps. Questions, do you have questions? Thank you for the talk, first of all. That was very interesting. Questions?
Come here, come on. Yeah, yes.
You were afraid to go and work with immunologists.
Yeah.
How did the immunologists react to you as a molecular biologist coming to them?
It depends on which phase of my study, our study.
In the beginning.
At the beginning, I was fired. I went there with a 2 years contract. And toward the end of second year, I went up to director, And I said, I can't stop the work I'm doing. Give me renewal. And he said, oh, okay, so I'll give you 1 more year. So she gave 1 more year. So I started going back to the lab, continue to work. And I was not really worried about my job for the future. But anyway, so toward the end of the 3rd year, I went back again. And he said, Susumu, I already extended your contract. That's it. I remember. That's it. That's what he said. So I was fired. But there was a time when the initial phase of work was going very well. Okay, I got a messenger RNA, I got the restriction enzyme, everything is ready. So I decided I should not listen to my director. I ignored it. I kept working with my few people I had together. And then by rumor, director found out what we are getting. So, not some way, I guess my contract was expired. Re-extended. Okay, but I never worried about it actually because I have this very extreme philosophy. If you are doing something worthwhile for human beings, nobody should stop it. Director, oh, they should not have such a power. But after that, when this was all worked out, and when he got to know exactly what we are finding, it was the opposite. He gave me as much space as I want and as much slot I want as a postdoc facility. So I went through that very drastic changes. But I don't blame him because he's a cellular immunologist. He doesn't really know anything about the molecular biology. So it's all right, but so that's the way it is. So I want to tell you, take this philosophy. If you are really doing a good thing, don't give up, okay? Don't give up. There will be a way. It will work out. It will work out. Even if the director said no, other people will not accept that. So you'll get the supporters. So don't get too nervous. Just keep doing good work. Yeah. That's my
The Swiss were always keeping you begging for money anyway. Yeah. Yes, please.
As a cell, we have those great kind of memory
Yeah.
and variety, so we can recognize many antigens. But you are doing neuroscience now, so I'm asking, why
Yeah.
other people, as a human, we cannot recognize human collectory and cannot memorize?
Okay, so I didn't say, I was meaning to say a little bit of a real memory study we do, but I cut it off because I didn't want to keep you guys too long. But the point is, after I moved to MIT, for a while I kept working on the immune system. In fact, the T-cell receptor and enhancer work was done at MIT. But it was still immunology system, immune system. But I'm beginning to realize that time is coming for me to change the field again. You know why? Because immunology is some of the question, remaining big question is very much medically oriented, okay? Well, you have to study on human patients, not mouse. So I thought these people who have a training, not only molecular biology, also the medical training, MD what is called the MD, PhD students. Most of them in America, they are usually very bright people. And I thought I should let them do it. And I'm going to start over in some other system, some unsolved problem, and try to see whether the eukaryotic molecular biology is still powerful enough to solve some of those problems in a nervous system. Okay? So it took some time to, again, to put them things together, but I focused on memory, learning and memory, because I had this very biased opinion that one of the most interesting thing about the brain is the fact that we have a memory. Okay? It's not, we are not being dictated by just the genes which we inherited from their parents. We have experience, our own private experience after when you grow up and after you grown up. And you use that information, keep it at your memory, not anybody else's, your memory. And you use that as the sort of a principle to decide what to do, what to choose, right? So I think the memory is very important problem in an important, powerful function of the brain. Okay, so if you're gonna try something new, why don't we try something most interesting? And therefore, I decided to try the memory system. Okay, and I was very fortunate. Around that time, I got a postdoc who came to my lab to work on the immune system. But one day, when we were talking over lunch, he said, and his name is someone called Alcino Silva, who is a professor now at UCLA. And he said he's real interested, although I'm doing immunology project, I'm really interested going into nervous system. So that coincided with what I wanted to do. And therefore, we agreed to try one project. And that's how we started. And in the following 5 years, all other postdoc graduate students, they also joined us. They switched from immunology to neuroscience. So, it's a question of what you think is interesting, because that's the most powerful drive to discover something new. I mean, so you have to decide what you want to do. And if you want to be a scientist, you have decided to be a scientist, okay, that's great, that's a good decision. And then try to find something which really excites you, and try to work for it. You don't have to work by yourself, but talk with the people, talk with not only the professors, but talk with the people around you, intelligent, smart people around you, friends, and test your idea of what one should pursue, what project one should pursue. Don't do it something just because it sounds interesting. It's not. There are so many things one can explore in science, and I wouldn't recommend when you're young. Try to fill in some details of something which is already going on. I wouldn't do that. You try to think about the impact, the big issue, something new, which is not being actively pursued by the people. Okay, and You'll be fine. You'll be fine if you're motivated. You'll be happy, making a little bit of progress. And that will give you more drive to continue and do even more riskier one. So at least that's the way I have lived my scientific life. Okay, and as I told you, even if I was fired, I decided I should not leave the lab. So, I mean, it works. It works. It's up to how motivated you are. If you are motivated, there will be a way to resolve a little small problem in front of you. That's the way. I mean, maybe my philosophy is too reckless. Maybe it's not applicable for everybody. And I'm not surprised it's not applicable to everybody. But that's the only way I have worked. Of course, there's a limit to it, because when you get older, my age, it's difficult to
It might be getting harder to find new things. Once you've moved to the brain,
another thing that I want to, if you most of you are students, so I'm not talking with the established scientists. One of my lessons is what I was trying to explain. That is, try to find some interesting question, unresolved question in the interface of science. You understand? You don't have to go to biology, to physics, but immunology versus molecular biology, yet close enough. And in the interface, there are problems which is unresolved, because neither of these people, they don't know what is available in the other side. They don't talk to each other, they are very specialized, most of them. So that's where there is a goal possible. The more solve the problem which is worth solving in the interface. Okay, so that's another thing you should think about. Don't stay in just in a field which you are used to, you are trained about. Try to explore. Even after you are finishing Ph.D., you can go to other field and try to actively train yourself in another field. And I have a postdoc who is right now, who is actually have a dual training in molecular biology and AI. Okay, artificial intelligence. I don't know anything about AI, so I can't advise anything to him. He teaches me what one could do in AI, and then sort of biological neuroscience and the AI is going to be a sort of a interactive area. Okay, so it's not for me, because my mathematical expertise is not very good. But younger people can think about the future, next 10 years. What will be exciting things going on then? I think you can't avoid thinking about some, I shouldn't say it that way, but I think you don't have to go directly only to AI. Something in the interface between neuroscience and the AI. These AI people, expert AI people, are now coming back to neuroscience. They come to ask how the human brain does this and that, because they want to use that, they want to borrow that information to see whether that can be applied to the model, in silico model. So I think if you are trained in molecular biology or molecular neuroscience or something, I think there is 10 years from now, that interface between those biological system and the in silico system modeling with the AI could be a very productive area. I don't know.
Okay, I think we better start to wrap up. Anyone want a last question? No. Yes? No. Okay. I think, thank you, Susumu. Very interesting. Yeah.